Joshua Williamson is a 5-year-old boy. He's brought to his pediatrician because he seems overly clumsy. Josh's mother states that he falls more than his playmates in daycare, and he was evaluated in the ER after taking a fall when he lost consciousness. The daycare workers indicate that Josh fell from the jungle gym. Josh's mother indicates that Josh can only run short distances without getting tired. Over the last few months he's been walking on his toes, and he's developed sort of a waddling gait. Josh's past medical history is unremarkable. He's lived with his adoptive parents since 2 months of age. Medical records for Josh's biological parents are not available. On physical exam, you note that Joshua shows decreased muscle tone, absent knee reflexes, and hyperreflexia at the ankles. When asked to stand from a supine position, Josh appears to use his hands to climb up his legs. His CBC is normal, blood chemistry is normal, except that serum creatinine kinase is elevated. An electromyography is abnormal. In today's presentation, I want to stress importance of interactions between membrane and cytoskeletal proteins, especially in maintaining the shape of cells. I would like to highlight a little bit the function of spectrin in the red blood cell membrane. We'll point out that dystrophin is a protein related to spectrin, and that the gene that codes for dystrophin is in the spectrin gene family. Now, if you haven't already figured it out, Josh has muscular dystrophy. So you should try to learn something about the genetics of the disease, maybe the chromosomal locus for the dystrophin gene, and maybe the location of dystrophin protein in skeletal muscle cells. From a clinical perspective, what are the symptoms of the disease? What's the age of onset for the disease? What are the treatments and prognosis for patients that have muscular dystrophy? Now, today we're going to be talking about membranes, so our learning objectives, you should be able to describe the biochemical components of membranes. You should be able to describe the fluid mosaic model for membrane structure. You should be able to describe some membrane cytoskeletal interactions, and you should be able to understand and appreciate the diverse function of membranes. In conjunction with what you had last term in biochemistry and what you're going to cover this term in physiology, you need to fully understand membrane structure and function, especially as this relates to transport, to receptors, and to signaling. Now, for the laboratory, you need to be at ease with the use of your microscope. There's a podcast that you can review to help you get familiar with the microscope, and then you're going to start the cytology lab this week. And there's a brief podcast on cytology overview. And at the end of the presentation, I will just show you a few slides to get you oriented to what you'll be seeing in the laboratory this week. You also need to understand and be able to explain the structure of the cell at all levels of organization. There's an introductory podcast on iTunes about that. There's also an audio podcast on iTunes that kind of reviews some cell biology, and then there's the CTB guts module just on kind of basic cell biology. So those are things you might review. Much of medicine is becoming genetic in its orientation, so I just highlight on this slide chromosomal loci for some genes that we may mention in our membrane discussion. I list the LDL receptor, the spectrin gene, and dystrophin. I am not going to test you in this course on these chromosomal locations, but I just include them for your reference. Now, what we're going to talk about today, I hope, is going to be review for most of you, but if you look at the cell membrane, I think we can agree that membranes would be critical for the origin of life and for the maintenance of life. Membranes envelop cells. They define a cell's shape and boundary. Plasma membrane certainly maintains distinctions between the inside of the cell and the outside of the cell, so it kind of separates the internal and external milieus of the the cell. Membranes control movements of material into the cell or out of the cell. They can act as passive barriers or as selective filters. As you know, membranes can detect external signals because there are receptors in membranes. Membranes then play an important role in cell-to-cell communication, and as you've already been exposed to in biochemistry, many signal transduction mechanisms start or are initiated as receptors bind something on the cell membrane. Membranes, as you are aware, serve as a locus for many diverse biochemical reactions, and as you'll appreciate in physiology, membranes can generate and maintain differences in ion concentrations either across the plasma membrane, so differences inside and outside of the cell, or across organelle membranes. Now, you should recall that there are three types of lipids that are components of all biological membranes: phospholipids, which are the most abundant, glycolipids, and cholesterol. Remember that lipids are amphipathic molecules, meaning they have a polar or a hydrophilic region and a nonpolar or a hydrophobic region. This is just an example of phospholipids. Here on the left side of the screen is just a schematic diagram, a chemical diagram in the middle, and a space-filling diagram on the far right of the screen. Remember that phospholipids can show different degrees of saturation, and they can have different length carbon side chains. And we can generally say that the greater the membrane fluidity, the shorter the side chains and the more unsaturated the side chains. Now, membrane fluidity, it turns out, is a fairly important concept. And what do we mean by membrane fluidity? We simply mean the ability of molecules to move around within a membrane. Now, this diagram highlights a classic experiment to show membrane fluidity. And what it's describing is basically a cell fusion experiment between a human cell and a mouse cell. And the experiment was set up so that antibodies are made against proteins in the human cell membrane, and antibodies are made against proteins in the mouse cell membrane. The antibodies to the mouse cell membrane would be labeled with a fluorescent probe like fluorescein, whereas the antibodies to the human proteins would be labeled with a fluorescent protein like, say, rhodamine. Now, under certain conditions, you can do what's called a cell fusion experiment so that the mouse and human cells can fuse. That forms what's called a heterokaryon, meaning a cell that has nuclei from two distinct sources. When that fusion is set up, at time 0, you have a cell, a heterokaryon, that has proteins that on one side are just mouse proteins, and on the other side are just human proteins. And if you were to stain and look at the fluorescence of this heterokaryon, you would have at time 0 a cell which is half one color, red for the human proteins, and half green for the mouse proteins. If you allow this cell to incubate for 30 to 40 minutes, you'll have a mixture of fluorescence, so you'll have just one color fluorescing back. And that's because the proteins within the membrane have moved around within the plane of the membrane. So this is membrane fluidity. Now, recall there are 4 major classes of phospholipids in plasma membranes, and these have a relatively asymmetric distribution. Within the interface of the plasma membrane, one finds phosphatidylethanolamine and phosphatidylserine. Within the outer face of the membrane, one finds phosphatidylcholine and sphingomyelin. Now, I'm not going to ask you to remember the chemical structure of these membrane phospholipids. They're all neutral phospholipids with the exception of phosphatidylserine, which is negatively charged. Now, lipids can move within an artificial lipid bilayer, and the movement of lipids within an artificial bilayer approximates what you see within a living cell membrane. So lipids can move laterally within one face of a membrane, so they can move around laterally within a membrane. Lipids can rotate about a long axis within a membrane. The lipids might bend or flex wherever there are unsaturated side chains, and lipids can actually change face from one face to the other face in a membrane. Now, in artificial bilayers, that is a rare event, but that actually does occur to a greater or lesser extent in living cell membranes, and we'll ask you to remember that for something that we'll bring up later in the course. Now, if one looks at distribution of carbohydrates across a membrane, this would be, for example, a lipid bilayer, and here on the bottom of the image would be the cytosolic face of the bilayer, and here on the top of the screen would be the extracellular face of the bilayer. These big green structures represent membrane proteins, and what this is showing is that carbohydrate groups that associate with membrane proteins or membrane lipids always point away from the cytosol. So they are on the extracellular side of a membrane. That is a almost general rule that sugar residues, carbohydrate residues, point away from the cytosol. Now, cholesterol is noted in that it maintains mechanical stability and fluidity of membranes. And here on the left side of the slide is just the chemical view of cholesterol, and you know that it's a planar structure. You can see kind of the steroid ring type structure. Here is just a schematic view to emphasize that planar ring structure, and then a space filling model. Because of the planar structure to cholesterol molecules, the molecule of cholesterol can kind of fit within phospholipids in a membrane, so you can get a more stiffened region of the membrane, phospholipids, and a more fluid region of the phospholipids. And it turns out that the amount of cholesterol within membranes can help to maintain the stability as well as the fluidity of membranes. Now, a point that I'd like you to remember, there is, and we already mentioned this before, an asymmetric distribution of phospholipids and glycolipids within the plasma membrane. Cholesterol is thought to be evenly distributed in both halves of a plasma membrane, but if you were to look at the extracellular portion of a plasma membrane, the phospholipids would be primarily phosphatidylcholine and sphingomyelin, whereas the cytosolic face of the membrane, the phospholipids would be mostly phosphatidylethanolamine and phosphatidylserine. Now, generally we say that biological membranes are pretty similar. In other words, they're conglomerations of phospholipids and proteins. Now, I've got a table here. I don't want you to really remember any specific things on this table. I just put it up to make a point. We're looking at, for example, membranes of, say, a liver cell, a red blood cell, a neuron, and then the average components of the inner and outer membranes of mitochondria, an ER membrane, and then kind of as a comparison, a membrane of an E. coli. And for example, going across, there's differences in cholesterol in these membranes, there's differences in some of the phospholipids in these membranes, or in some of the other glycolipids. So while one will generally say that all biological membranes are conglomerates of proteins and various phospholipids, there are distinctions from one membrane to another, and some of those distinctions turn out to be important in defining the chemical differences and functions of membranes. And that's the take-home message I want you to take from this slide. Another little bit of review for some things. If you take lipid molecules and have them within water. We know that because lipids and phospholipids have polar and nonpolar regions, they can spontaneously form lipid micelles and bilayers within an aqueous solution. And that's simply from the thermodynamic properties of the phospholipid molecules and the aqueous environment. These lipid bilayers can readily form liposomes in an aqueous solution, and this liposome almost looks like a hypothetical model cell. And indeed, people have experimentally transfected cells by putting substances in the lumen, if you want, of this liposome, and because these are phospholipid molecules, these can be readily soluble in the plasma membrane of a cell. So you can actually transfect a cell by putting material into the lumen of these liposomes. This is a schematic representation of a lipid bilayer, and this is something that everybody has learned from elementary biology. If you freeze fracture a biological membrane, it's going to split at the point of least resistance, which will be right down the middle of the lipid bilayer. These green globules would be protein molecules that are spanning the lipid bilayer. If you were to do this freeze fracture and heavy metal shadow the preparation and look at it under the electron microscope, you'd see an image like that's shown on the bottom, where you'd see little bumps and ridges. This represents the fact that a lipid bilayer is really a two-dimensional fluid. These proteins float freely in the plane of a lipid bilayer. That is membrane fluidity. Now, it turns out that as cells organize into tissues, various mobility restraint mechanisms have to operate to limit the mobility of proteins within the membrane. In other words, to limit the fluidity of membranes. And this is an interesting example. This is a schematic or a hypothetical example of a mobility restraint mechanism. Here I'm just calling it a tight junction between cells that might limit movement of proteins, shown either as green little lollipops on the cartoon or red little lollipops. In this case, the apical plasma membrane domains are defined here on one side of the tight junction, and the basal and lateral or basolateral membrane domains are shown on this side of the tight junction. And in the cartoon, the implication is that these proteins that are kind of shown as the red lollipops can float freely within the basal lateral domains of the membranes, but they can't cross the tight junction. And likewise, these proteins that are labeled as green lollipops can float freely in the apical membrane domain, but they can't cross the tight junction and get to the lateral or basal domain. Now, the cartoon on the right depicts reality for intestinal epithelial cells, and you probably can appreciate that there's a high concentration of glucose within intestinal epithelial cells. And you can probably appreciate that there's something called the sodium-potassium ATPase, which is on the basal domain of these epithelial cells. This sodium-potassium ATPase functions to maintain a high concentration of sodium outside the cell and a relatively low concentration of sodium inside the cell. Well, in the apical plasma membrane domains of intestinal epithelial cells, there is a sodium glucose symporter. And what this means is that sodium, which is in high concentration outside the cell, can flow into the cell on this carrier protein, and this carrier protein will also bind glucose. So glucose can move against a concentration gradient going from the apical or luminal side of the epithelial cells into the cell. And then that glucose in high concentration can be shuttled down its concentration gradient on a glucose transporter, which is located in the basal side of the cell. So the glucose can get from the cell here into the extracellular space and eventually into the blood capillaries. So in this case, the sodium glucose symporter is confined to the apical domains of the cell, whereas the basal domain is confining the sodium potassium ATPase as well as this passive glucose transporter. Here is a histological view of the intestinal epithelium. For right now, don't worry about these blue cells. They're goblet cells. Here are the absorptive cells. They're tall, simple columnar cells, which we'll talk about in a later lecture. But the absorption of glucose is occurring in this region, and then it's going to be transported through the cell to the basal region, where the glucose will be excreted into the blood capillaries. This is an electron micrograph of the area we just saw. So this is the luminal surface. Lots of microvilli on the cell surface with an adsorbed glycocalyx. This is one cell. This is another cell. Here is the tight junction, a so-called zonula occludens. Here is a zonula adherens and a macula adherens. Macula adherens, also called the desmosome, and the zonular adherens we'll talk about in more detail next time. Here I'm just highlighting the zonula occludens, or tight junction, and just want to point out that anything in these membranes is free to diffuse or move all the way through this region of the membrane, but not able to pass that tight junction. Okay, and likewise, any molecules that are in the membranes here are free to diffuse all through these regions of the membrane, but can't pass this tight junction. So anything in the basal lateral regions here cannot get in the apical region here because of not being able to pass the tight junction, and likewise anything in the apical regions here can't get in the basal or lateral regions here because the proteins here can't pass that tight junction. This is just a little bit higher power view of the junctional complexes here. And again, we're going to come back to this at a later lecture, so I'm going to not go into any more details right now. Now, proteins can associate with the lipid bilayer in several different ways. We can talk about what are called integral membrane proteins and peripheral membrane proteins. And the first thing to think about is an integral membrane protein is a protein that is intimately associated with the plasma membrane. It may span the plasma membrane one time or multiple times, usually crossing the membrane in an alpha helical configuration, although you'll recall from biochemistry there are some other configurations that proteins can assume as they cross membranes. But these integral proteins are very difficult to dissociate from the membrane. If you wanted to study them biochemically, you'd have to actually rupture the cell and with very harsh detergents solubilize the membrane to isolate these integral membrane proteins. On the other hand, peripheral membrane proteins are relatively easy to dissociate from the membrane. Here we see a peripheral membrane protein on the cytosolic face of a plasma membrane, maybe just weakly associated with some type of a carbohydrate side chain. Here, a peripheral membrane protein on the extracellular side of the plasma membrane, and then here a peripheral membrane protein on the cytosolic side, a peripheral protein on the extracellular side. In this case, maybe it's a cytoskeletal protein interacting with a membrane protein. In this case, maybe an extracellular protein interacting with a membrane protein. So this is going to foreshadow a membrane protein cytoskeletal interaction. Here, a membrane protein extracellular matrix interaction. This latter very important when we talk about connective tissue. Now, what I wanted to do is give a brief clinical correlation and talk about membrane fluidity, cholesterol uptake and utilization, and a little bit of genetics. What we're going to consider is the ability of individuals to bind LDL and to internalize LDL. And we can consider this in individuals that are normal with respect to binding and internalization of LDL. And we can consider this with respect to individuals that have hypercholesterolemia. Now, you should appreciate that LDL transports cholesterol and triglycerides from the liver to peripheral tissues in the body, and high levels of plasma LDL can correlate with arteriosclerosis and cardiovascular disease. An LDL particle is about 3 million Daltons in size. It's essentially 1,500 cholesterol molecules that are esterified to fatty acids. The LDL particles are surrounded by a lipid monolayer, and this is organized into a fairly large single apoprotein. LDL, you may recall, originates in the liver, and you should realize that LDL is given the name bad cholesterol. And that's kind of a schematic of what the LDL particle looks like. Well, this is how LDL is internalized into the cell. Here's the LDL particle, would have been this particle. So here's the LDL particle in extracellular space. Here's the cell membrane. And here are specific LDL receptors in the plasma membrane. It turns out that when this receptor comes across its ligand, called the LDL particle in this case, these receptors will cluster into what's called a coated pit. The coated pit will pinch off into a coated vesicle. This coated vesicle will get internalized into the cell. It will ultimately lose its coating. It will fuse with an endosome, and in the endosome, a component will bud off and recycle the LDL receptor in a vesicle back to the cell surface, whereas other regions of the vesicle will bud and fuse with lysosomes to digest the LDL into free cholesterol. This LDL internalization occurs by something called receptor-mediated endocytosis. And it turns out that the lifespan for the LDL receptor is about 20 hours, and one cycle of this internalization, whereas the LDL binds to the receptor is internalized and recycled back to the cell surface, one cycle of that takes about 10 minutes. Now, here is the LDL receptor, and it's shown with several functional domains. So there's an LDL binding site domain, and there's a coated pit binding site domain. Now, I have not talked about what makes up this coated pit, but it is essentially consists of molecules of clathrin and other coat-associated proteins. And you have to have the interaction of the LDL with the receptor and the receptor with these coated pit type proteins to actually allow internalization of LDL to occur. Here is an example of a mutation in the LDL receptor such that you don't have a functioning part of the LDL domain that binds with the coated pit region, so you don't have normal internalization. that can explain exactly what you see in these data. Here would be normal LDL binding, normal LDL internalization from our normal patients. Here you would have normal LDL binding because the receptor has a normal LDL binding site, but you don't have normal internalization because the receptor has a mutation in the LDL internalization site. So that type of mutation can explain these data. If you were to look at these data, you might envision that there'd be a mutation in this region of the receptor so that you don't bind LDL. Hence, if you don't bind LDL, there can be no LDL internalization. What this means is that hypercholesterolemia can be caused by several classes of mutations that relate to the LDL receptor. So, for example, you may have a mutation in part of the LDL receptor that's involved in its original synthesis in the endoplasmic reticulum. You may have a mutation in the receptor that's involved in the transport of the receptor from the endoplasmic reticulum to the Golgi, or maybe a mutation in its transport through the Golgi. There could potentially be a receptor mutation that's involved with LDL binding or with LDL clustering or internalization, which with the mutation that we showed in our example, or maybe a mutation in the recycling of the LDL back to the cell surface. Any one of these mutations could lead to hypercholesterolemia. It turns out that familial hypercholesterolemia are due to these mutations in the LDL receptor. This is inherited in a Mendelian manner. LDL gene is on the short arm of chromosome 19. LDL receptor mutations are one of the most common inherited disorders in humans. It's usually an autosomal dominant condition if it's a receptor defect. About 1 in 500 individuals have one defective LDL gene, and those individuals will begin to show coronary artery disease or cardiovascular disease by somewhere between, like, say, the third and fourth decade of life. If you're one of the unfortunate individuals that carries two defective LDL genes, that's usually about like 1 in a million, those individuals usually don't survive much into the second decade of life. Hypercholesterolemia. What are the signs of hypercholesterolemia? Fatty skin deposits or xanthomas that you can see in various parts of the body. A family history, especially of early heart disease or high LDL levels. Blood chemistry. Total cholesterol might be greater than 300. LDL might exceed 220. And it's important to know what these values are in children if you're diagnosing somebody relatively young. And interestingly, triglycerides can generally be normal with people with hypercholesterolemia. So those might be the signs. Symptoms: you might have a chest pain type symptom, angina, or other symptoms of vascular disease. Treatment for hypercholesterolemia might be lifestyle changes, might try to have fat intake to be about less than 30% of the total caloric intake, perhaps might reduce meat intake or dairy intake, may reduce intake of certain oils, and obviously there could be pharmacological intervention with drugs like statins. Now what I'm showing here are red blood cells, and this is a scanning electron micrograph of red blood cells, and what's very characteristic of these cells are the biconcave shape. The biconcave shape of red blood cells is due to interactions between membrane proteins and cytoskeletal proteins. Now, let's make a couple of comments about red blood cells. First of all, they are essentially bags of hemoglobin, and you can take red blood cells and rupture the red blood cells and create what are called red blood cell ghosts. And this is basically just a bag of membrane which can be useful for studying membrane structure function. And under certain conditions, you can take a red blood cell and lyse the cell and make a sealed right side out vesicle or a sealed inside out vesicle. These sealed inside-out vesicles turns out are relatively useful for studying the structure of the inner face of the red cell membrane and the red cell cytoskeleton. If you were to take these sealed inside-out red cells and heavy metal shadow them and look at them under the electron microscope, you see a structure, a very organized structure. And we've come to appreciate that that organized structure represents the spectrin-based membrane cytoskeletal interactions that are important in maintaining the shape of a red blood cell. It turns out that in this interaction there are spectrin dimers that associate head-to-head to form tetramers with each other, and they form tetramers in conjunction with actin and tropomyosin, which you might recognize as cytoskeletal proteins. They also interact with proteins called band 4.1 protein and adduxin. Band 4.1 protein is just named by virtue of how far it migrates on an electrophoretic gel. These spectrins also bind by virtue of ankyrin to band 3 protein, another cytoskeletal protein. The take-home message here is this interaction between the membrane proteins, like the band 3 protein, and the cytoskeletal proteins like the spectrins, the ankyins, the actin. These interactions are important to maintain the shape, the biconcave shape of the red blood cell. Now, here is a little more complex diagram, not of a red blood cell, but just of a hypothetical epithelial cell, and it's showing the cortical region of the cell. That is the region of the cell shown on the top near the plasma membrane. Here is the lipid bilayer, and it's showing integral membrane proteins. Here they're alpha and beta subunits of integrin that are interacting with cytoskeletal proteins like alpha-actinin or vinculin or talin. Here the alpha-actinin interacting with F-actin, and here talin interacting with F-actin and vinculin, showing a complex interaction of cytoskeletal proteins and membrane proteins. Here on the extracellular side, showing interactions of the integral membrane proteins integrin with extracellular matrix proteins like fibronectin or laminin. So again, showing interactions with membrane extracellular proteins, which are very important in connective tissue function, or membrane cytoskeletal proteins, which are very important in maintaining structural integrity and function of all kinds of cells. Now, here is spectrin, and it's in a striated muscle. I don't expect you to remember anything on this complex diagram except its take-home message that spectrin is related to dystrophin, another cytoskeletal type protein. You can imagine mutations in spectrin that might disrupt the membrane cytoskeletal interaction of striated muscle, or look at a mutation in dystrophin might disrupt the membrane cytoskeletal interactions in the muscle cell, and this mutation in dystrophin could cause something like muscular dystrophy. Say a few quick words about transport across membranes. You're going to cover this in much greater detail in your physiology course, but you know that transport across membranes might be passive transport or active transport, whether something is going with a concentration gradient or against a concentration gradient. I think you can appreciate that these concentration gradients can be established to do work within cells. I think you might be able to appreciate that material can move across a plasma membrane by simple diffusion if the molecule is soluble in the plasma membrane. If it's not soluble, it might move across the membrane by a facilitated diffusion, either through a channel or with a carrier protein. This is all passive transport because the molecule is moving with a concentration gradient, either by simple diffusion, by channel-mediated diffusion, or by carrier-mediated diffusion, the latter two being examples of facilitated diffusion. On the other hand, a molecule might move against a concentration gradient with a carrier protein, and that would require energy moving against a concentration gradient. That would be active transport. Now, we know that molecules cross membranes in various ways, and so that ion concentrations can be established across membranes. So, for example, there is a much higher extracellular concentration of sodium compared to its intracellular concentration. The concentration of chloride follows that of sodium. On the other hand, there's a much higher intracellular concentration of potassium as compared to extracellular concentration. And you can see there's more magnesium intracellular than extracellular. So the point being that there are differences in ion concentrations across a typical plasma cell, a typical plasma membrane of a cell. I'm sorry, I didn't mean to say plasma cell. Now, material can also cross membranes by a process called exocytosis, where you've got material within a vesicle. This vesicle will fuse with the plasma membrane and material can be released from the cell, or kind of the opposite, endocytosis. A vesicle might pinch off and bring in some material that's outside the cell into the cell. We saw an example of that with LDL internalization, and we're going to talk later about budding, whereas a vesicle might bud off of an internal membrane and go to another area within the cell, fuse with that area in the cell to transport something from one organelle to another, or you might imagine this vesicle budding off and fusing with the plasma membrane for exocytosis. We can talk about the fact that most membrane compartments are related by concepts of membrane flow. This is a complex slide. It has a lot of information on it, and I'm going to leave it for now because it's going to lead into something we'll talk about later in the course. But I just want you to get a general appreciation of the take-home message that material might flow by membrane flow from one region of the cell through another region of the cell to the cell surface. Likewise, things might be internalized by membrane movement. So we're going to talk about this in much more detail later. For now, don't worry about default pathways or regulated pathways. We'll talk about that later in the course. So let me just summarize what we've said about membrane structure and function. Lipid bilayers provide the structural framework for all membranes, whether we're talking about the plasma membrane or different organelle membranes. Now, we generally think that although membranes are similar, there are differences, of course, in chemical composition between membranes. Those differences are going to reflect differences in either cell type or in function of membranes, whether you're talking about organelle membranes or membrane function from one cell to another. Membranes are fluid. There can be two-dimensional movement of molecules within membranes because they're fluid mosaic structures. Membrane proteins can move freely in membranes. Membrane lipids can move freely in membranes. But as we've shown, there'll be situations where you have to limit movement of material within membranes. Proteins of the plasma membrane interact with proteins of the cytoskeleton or with proteins of the extracellular matrix. We'll come back to that later in the course. Proteins will mediate all kinds of membrane function, whether it's cell-to-cell recognition, whether it's cell signaling, whether it's transport of material across membranes, or even as sites of metabolic activity. and we suggested that membranes are asymmetrical with respect to their composition and hence their function. Now, let me just give you a brief overview of some slides that you're going to see in the laboratory. And you're going to be doing this over the next several days. So this is just serving as an introduction to some slides that you'll see. First of all, we show here a neuron. And here you can see the nucleus and the nucleolus. Okay, and then a very darkly staining cytoplasm of the neuron. This cytoplasm is kind of lumpy or clumped in appearance. This is due to the Nissl substance, which is rough ER. Here are dendrites coming in to the cell body on this neuron, and right here is the region called the axon hillock. This remains unstained because there's no rough ER extending into the axon of the neuron, and you'll appreciate this as we talk about nerve cells later. Here's again a multipolar neuron. You can barely see the nucleus here, the cell body. You can see some dendrites coming in, and this is probably the region of the axon hillock, where the axon is coming out of this neuron. Here, some smooth muscle, very characteristic fusiform or spindle shape of smooth muscle. You can see the central nuclei in these cells. And we're going to talk about, obviously, nerve cells and muscle cells in much greater detail later in the course. Here's a capillary, basically consisting of portions of one cell that I'm tracing here, like this, and another cell here. This cell has the region of the nucleus in the plane of section. This cell, on the other hand, does not. Here's the capillary lumen, not much bigger than to allow a red blood cell to fit in that lumen. The next slide shows a higher power view here, showing junctional complexes between the two endothelial cells that make up this capillary. And we'll talk about these structures in more detail a little bit later in the course. Here's the slide we showed earlier of the GI epithelium. So here's the luminal space of the GI epithelium. So the apical regions where absorption occurs. These blue cells are goblet cells. We'll talk about these again a little bit later in the course. This is just a high power view of this region, for example. And you can see the portion of one cell, a second cell, a third cell, and a fourth cell. And in this GI epithelium, you can see the apical surface elaborated into microvilli. You can see junctional complexes between this cell and that cell, or between that cell and that cell. This elaboration of microvilli is essentially increasing the luminal surface area for absorption, because remember, this region corresponds to this region, the luminal region where absorption is occurring. I would never expect that you would recognize that this rough ER and mitochondria are from the pancreas. I just show this to show you a high power view of the mitochondria. You can see mitochondria as double membrane structures, then outer membrane and inner membrane folded into cristae, mitochondrial matrix, the rough ER with ribosomes associated with it. Within the mitochondria, you can see these electron dense structures. These are actually calcium deposits. Here is a view of spinal ganglion, and it's with a specialized silver stain to highlight the Golgi apparatus. Nucleus, nucleolus, and the Golgi apparatus, staining with a special heavy metal stain. And then a high power view of the Golgi apparatus in a cell like what we've just shown. You can see the Golgi here with a characteristic structure, and you can see the ER with its characteristic structure. You will need to review some of the cytology to appreciate some of the structures of the different organelles. And this should be familiar to what you've covered last semester in biochemistry.